I just want to remind everybody that this issue has just come out. Joan Lunny and myself, along with XJ Ming, on alternative strategies to PRRS. Some of the topics we're talking about today are featured in that issue. Just a reminder to anybody who does PRRS, there's sort of been a little bit of reorganization in terms of the family Arteriviridae. In the past, we've always looked at this group of Rodarteriviruses, but now the situation has changed, and really we are just focusing on this group of viruses within the Rodarterivirus group. This is very important because this new reorganization of the nomenclature has been instrumental in really dividing these viruses into different functional groups in terms of how they function immunologically and also for the development of vaccines. Also, probably a lot of you people in this room who do PRRS, I review your manuscripts. Make sure in the introduction you have this new nomenclature. If it's the old nomenclature, you're going to hear something from me. So when we look at PRRS, this is how we see it at the population level. And these are pictures that we built by infecting 200 pigs at a time. And the most sensitive instrument that we can use to measure PRRS is a scale by just looking at weights. If you take a group of infected pigs over time and you look at their weights, you can see that the population structure just falls apart. And you find these pigs at the very end, and these are called laggers. It costs money to bring those laggers up to the front. And so I promised Eric Newman I would keep a commitment to him. He made this picture back in 2003, which really describes the persistence of PRRS within a production system, as well as other ways in which the virus can get in from the outside. And this has really been one of the principal issues related to this virus. I always call it the big bugaboo, and that's really is persistence. Persistence within a pig and persistence within a population. These are PCR results for 200 pigs. It's not a very clean picture in which you see the virus disappear. It just keeps coming back, and in fact what we see is that in these large populations of pigs we actually see waves of virus reappearing in the emergence of escape variants. And so when we look at PRRS, well, what's its greatest cost? Being in a vet school, we always say health. But at the end of the day, it's really a waste in feed. It's a nutrition issue in which 10% or so of the corn or whatever is fed to pigs is lost, either due to sick or dead pigs, pigs that are slow performing in secondary infections. So when we look at the bigger picture, we see that PRRS is not just a health disease problem, but it relates to nutrition, environment, and also has some social impacts in terms of this feed that's wasted on the disease. I think what has emerged in the PRRS community is this idea of taking an integrated or holistic approach. For a while, we've had these more traditional approaches: vaccines, detection, epidemiology, biosecurity. Now we see people studying the nutrition. What's the impact of PRRS on nutrition, and how do you best feed a PRRS-infected pig? But then also some of the work done by Megan Nerdewerder and others looking at the microbiome. How is the microbiome composition important in terms of the PRRS response, and how does PRRS affect the microbiome composition? And then finally, the topic today is going to be on genetics. And it's not that easy because classically we think in terms of resistance. We're going to make this resistant pig the same way you make a resistant variety of corn. Well, it's not quite that easy. And we'll talk a little bit about what we mean by resistance. But nowadays, I think what you're going to see today is a discussion of tolerance versus resilience. These seems to be the hot button issues in terms of genetics. And then finally, this idea of having this vaccine-ready pig. What can genetics tell us about the vaccine response? And so just an overview of PRRS in this new genus we call Rodarteriviruses. The PRRS genome is divided into two parts. About 3/5 of it is devoted to the non-structural proteins. And then on the 3' end, we have this complex mixture of structural protein genes. This is one thing unique about PRRS. It's a relatively short genome, but it has a relatively complex surface structure. And when we look at PRRS in the surface structure, you can find all kinds of interactions between host and viral proteins that have been reported in the literature. So all of these proteins are somehow interacting with host cell proteins. Just recently, more recently, Kay Fauberg has found a non-structural protein that's a component of the PRRS virion. So this is a very complex situation. Probably what is known is that CD163 is required. And so the last part of my talk will just be devoted to a few slides regarding CD163. PRRS vaccines, we know all about them. We've had modified live vaccines since 1994. New vaccines coming on the market are some of the same modified vaccine designs. Where is the next generation of vaccines? Where are they and when are they? It's anybody's guess. And the question is will there ever be a perfect vaccine for PRRS? And so out of that, in about 2003, 2007, was can we really apply genetics to improve the health situation of pigs? And what I'll be talking mostly about is some of the ideas that, well, if we can find markers, genomic markers, then we can conduct things such as marker-selected breeding programs that would help improve the response of pig to the infection. But then also we're moving, of course, into that age of genetic modification. And if anybody saw Allison's talk last night, it was really devoted to what genetic modification can do for the industry. And so we see it really two ways. We can insert new genes or we can insert alleles that provide some benefit. But we can also delete out things such as receptors. And so I'm going to be finishing my talk talking about the deletion of CD163. And so again, 2007, as Jack said, is the formation of this consortium. And the idea is to develop these marker-selected tools that we can find pigs that are resistant to PRRS, pigs that may be tolerant or resistant. And again, this idea, can we identify a vaccine-ready pig? But then I think sometimes we kind of think in terms of resistance only, but don't realize that these are the same tools that we can use to identify novel pathways that provide a host defense. How can we integrate the identification of these novel pathways into the next generation of vaccines? And I guess what we're aware of every day is that I have about 10-80 freezers that are filled with nothing but samples. And so one thing that came out of this was just this huge repository of samples and tissues from this study, along with the repository of data. Anytime you see a pig, think of $1,000. In our studies, we run about 5,000 pigs. So we have contributed $5 million to just infecting pigs and collecting samples. That's a huge effort, and I always say we will never come this way again. We will never do this experiment again. And so when we look at models that we've used in the PHGC, the first model we start out with is a standard nursery pig model, and I give you some idea. We've run about, I think, more than 3,200 pigs through this model. Also, Jack mentioned some of the field models. Well, we can put them in the lab, but what happens if we take these pigs out to the field? And then this dual infection PCV2 PRRS disease model. One thing to keep in mind that every pig that you see in this study has been genotyped. And then also in terms of the nursery pig model, at least, we use well-characterized PRRS isolates. And at the end of the day, through Jack's work, we see the genome-wide association studies being done. You'll see mostly Jack's data, unless you want to see 1/2 hour of my lab bleeding pigs, because that's basically our contribution to the study. So I'll be showing a lot of the work that's come out of the consortium, but not necessarily from my lab. So this is the standard nursery pig model that we use, and I think anybody who does PRRS probably uses the same model. You start with a 3-week-old pig, you acclimate the pig, you infect it for 42 days. You collect a lot of samples during the first week because that's when you see acute infection, and then weekly thereafter. During that 42 days, we're looking at 2 aspects of PRRS infection. We're looking at the acute phase, or the acute replication, which occurs within about the first 28 days, followed by something we call persistence. Persistence in PRRS is poorly defined, but for the purpose of these experiments, if we find PRRS in the tonsil at 42 days, but not in the blood or the serum, then we call that quote-unquote persistence. Phenotypic data types, in terms of the samples that we collect, of course we collect serum for virus, cytokines, antibody, whole blood for peripheral blood monocytes. We've done quite a peripheral blood mononuclear cells. We've done quite a few of those studies. Of course, we can't do that on every pig. That's a huge amount of work. All the blood samples, just about all the blood samples that were collected during the study were stored as RNA, preserved in Tempest tubes. And you're going to see a lot of RNA-seq data that comes from these studies. Oral fluids, not that those are important, but we take those oral fluid samples and diagnostic folks use those to develop oral fluid tests. The latest thing is feces, to look at what effect PRRS has on calorie conversion in pigs, but then more recently on the microbiome. And just an editorial observation, a veterinarian would rather wrestle and bleed a 250-pound pig than collect a fecal sample. There's just something, loss of dignity when you have to collect fecal samples. So, but we have done a lot of those. And then again, understanding what happens in the tonsil as a means to understand persistence, as well as other tissues. Oftentimes, someone will call me up and say, hey, I understand you're going to do a study, can you get us this? And then we'll collect the samples for them. In types of the data that we collect, one thing that we wanted to make sure was that everything is reproducible. And so we oftentimes use a commercial kit. And that's what we use for our PRRS PCR, or we use standard diagnostic tests whenever possible. And this allows us to make this data transferable. We can transfer it between studies, but we can also transfer it between research groups. If there's someone in China or some other country who uses the same model and gets the same data, then there creates an opportunity to pool these data together into larger studies. Phenotypic data has got to be cost effective. You can't be spending all your money just collecting phenotypes. And that's one thing we found about weighing pigs. It's very cheap, and it gives you a very nice quantitative reproducible number. But then when we look at things like commercial tests, then we see a little bit more of an expense. One thing we've done during these studies, the phenotypic data that we collect is non-invasive. In other words, we don't look at lung lesions because you can't take the lung out of a pig, put it back in the pig, and put it back in the study. So there's a lot of things related to PRRS that we don't do, and those things are just obvious. Types of data, some of it is binary. Is the pig, we ask the question, is that pig alive or dead? And the one thing about the genomics people like Jack, one thing I learned is that if I give Jack a number, if I can give anything a number, he can manipulate it. He can turn it into a marker on the genome. So when we look at this binary data, it's plus minus. We also, to some degree, look at qualitative data. This is more hardly to work with because things like clinical scores are a little subjective. But I'll talk about clinical scores in a moment when we look at disease phenotypes. And that's, again, some of the work that was done by Megan Nerderwerder originally. But the best phenotypic data is data that is quantitative. And so we can measure things like weight gain, virus load, antibody levels. And these seems to be the best types of data, the best type of data for these types of studies. So here's our nursery pig model. We call this deep phenotyping. Morbidity, mortality, viremia, virus load, weight gain, antibody, total antibody, virus neutralizing activity, cytokine levels, virus level, and tonsil as a means of persistence. But from this cursory phenotypic analysis, we've since moved on to what we called ultra-deep phenotyping. And that is what happens when you look at the transcriptome. When you begin to analyze the transcriptome, the host transcriptome, you can give that stuff numbers. And again, that's the kind of thing that Jack needs to do as analysis. People always ask us, well, you can use one virus, does it work for another virus? So we sort of anticipated this, and in these studies, in the nursery pig model, we use two viruses. One is NVSL, 97, kind of a standard high pathogenic isolate, and KS206. And this is some very nice work coming out of Jack's lab. Andrew Hess did this work, and you can see that the two viruses that we use have two different properties. NVSL comes up very rapidly to a high level in the blood, and then decays fairly rapidly. KS06 is a virus which is what we call a little more persistent. It's a virus that kind of percolates. The viremic levels are lower. So we have these two different viruses that we put in these studies. Viral load. This is really cool because regardless of when a virus peaks or how long it goes, we can get an area under the curve value that we call virus load. What's interesting about virus load is we only take it to the first 21 days because we have this issue about reactivation in about 20% of the animals. So all the virus load data is really viremia, area under the curve, during the first 21 days. Pigs. We start with 200 pigs in each one of these trials. The only thing that I want to say is that we pick a population of pigs in which we can anticipate lots of genetic variation. The most important aspect to this is that there's no pre-selection for any PRRS-associated response. In other words, these are just production pigs in which we have a large variation in genetics within the population. Again, some of the data that comes out of this figure I showed you before. We also see weight gain here, and then viremia with this part of viremia that we call rebound. This is very interesting because we have seen this ever since the beginning of PRRS. Within the population, you see this huge variation in weight gain, variation in viremia, and in this distribution. This variation within the population of pigs infected with the same virus strongly suggests that there must be an underlying host component, again, a genetic component. And so from this, the first work coming out of Jack's lab is the fact that, a response to PRRS is really what we'd call a moderately inherited trait. And I says moderately, well, if you're a geneticist, moderately means a lot. Okay, and so that's a huge effect. And it's estimated about 40% of how a pig responds to PRRS is related to the pig and not the virus. But then we can look at other aspects such as the virus, environment, maternal effects, and whatever. And the impact of this early work was that this provided the strong suggestion that you can actually breed pigs for improved response to PRRS. And out of this is probably one of the most famous figures to ever come out of this study. This is what is called a Manhattan plot. It's called a Manhattan plot because normally it looks like the Manhattan skyline. Well, this is the Manhattan, Kansas skyline. Wheat field, telephone pole, wheat field, telephone pole. This picture says a lot. It said, number one, that maybe disease resistance is not that complex. We can map it just to a few areas on the genome. And the other thing is that using these experimental studies, look at this. We've taken that background Manhattan skyline and basically flattened it into a Kansas wheat field. So out of this came we have these two phenotypes, virus load and weight gain, and guess what? They converge at the same marker on chromosome 4. And this we call the chromosome 4 marker, or in short, we call it WUR. And this is from the first work done by Nick Boddicker. This is a horrible, long paper to get accepted for publication because this is the first paper to really look at the genetics of disease resistance. You send it to a disease person, they say, I don't know anything about genetics. You send it to a genetics person, they know nothing about disease. So it took a long time for this paper to become accepted. And so what does this gain, this WR genotype, what does it gain? If you have the favorable genotype, you can expect about 10% increase in weight gain. If you look at viremia, you look at a corresponding reduction in viremia. By no means this is resistance. But if you look at from the very beginning what PRRS takes away from us, I always say PRRS takes away 10%. Guess what? All we need to do is get that 10% back. We don't need a perfectly resistant pig. We just need something that's going to we can get back from the virus what the virus takes from us. And so where does this WR SNP localize? Some very nice work done by Jim Reese. Came right to this protein that is called guanylate binding protein 5. In terms of the gene, it looks like that the unfavorable genotypes have this premature stop codon as a result of these splice variations in the gene. As it turns out, guanylate binding protein 5 is very interesting. It belongs to a family of proteins that are upregulated through stimulation through gamma interferon. In fact, this was the first gene to be identified to be upregulated in response to treatment of cells with interferon. And so when we look at what GP, guanylate binding protein 5, it sits in the Golgi, it's anchored in the Golgi, and guess what? Where does PRRS replicate? At that Golgi-ER interface. when we look at what this protein does when it's upregulated in response to interferon, it's a marker for M1 macrophages, and it probably drives the differentiation of these macrophages. These macrophages are non-permissive for PRRS. PRRS infects this population of M2 macrophages that are CD163 positive. Through the formation of inflammasomes, the release of inflammatory cytokines, through stimulation of NF-kappa B pathways, induction of antiviral states. And then really interesting coming out of work done by HIV is that this protein may physically interfere with the replication or assembly of the virus itself. So that's the GP guanylate binding 5 story. I want to move on and say, okay, we've also taken these pigs out in the field, again, starting at 3 weeks and taking them all the way to market, starting out with 200 pigs in a barn, and then 50 pigs later on when they get too big. And what we do is we take these 200 pigs and we put them in a 1,000 hog barn that is diseased challenged. In other words, we look for barns that are funky. They have everything. And so we want to see how these pigs behave in the real world. And then the dual infection model that Jack talked about. This is really a disease model. And if it's going to be a disease model, we need something to describe disease. And this is where we looked at clinical signs. In this model, we start with 50 pigs that are of the unfavorable genotype. Or we start with 100. Half of them are non-vaccinated. The other one's vaccinated. We start with 100 of the favorable genotype pigs. Again, half of them vaccinated and half of them non-vaccinated. The vaccine we're using is a modified live PRRS virus. This is the results you get. If you have if you're not vaccinated, you see PRRS. If you're vaccinated, you don't see PRRS. Okay, good, the vaccine works. But later on down the road, when we look at PCVAD, if you're vaccinated, PCVAD is worse. And the reason is this is a live modified virus, more than likely functions the way PRRS normally functions during PCVAD by increasing PCV2 replication. So here, under these circumstances, yeah, having some clinical parameters and giving Jack a clinical parameter number is something that he can use to look at, well, what is the genetics of disease? And just some very recent work, again coming out of Jack's lab from Janelle Dunkelberger. Where does this virus load for PCV2, what is it associated? It looks like it's associated with something going on in chromosome 7. So I just want to end up with some of the work that's actually coming out of my lab. In 2 years ago, we described the use of CD163 knockout pigs and the fact that they are totally resistant to PRRS. And so the question is, so you got a pig in which CD163 is knocked out. That doesn't have a lot of utility, primarily because I like a little CD163 in my bacon. You know, I think it would, you know. So you really can't eliminate CD163 because it is important. So the question is, but how can we use it for PRRS? And this is reproductive PRRS at about 90 days gestation if the sow is infected. And these are fetuses that were taken out like at 109. And it's really, since we've already had lunch, it's really a smorgasbord of disease pathology. Pigs that look normal, pigs that are dead, pigs that are meconium stained. So it's just a smorgasbord of pathology. And so this is the experiment. The question we wanted to ask is, if we have a CD163 knockout dam, can it protect wild-type fetuses? This is the experiment, and it's just based on simple Mendelian genetics. Here you have a positive male, positive dam, and all the fetuses are positive. Here you have a positive boar, and if you breed that with a knockout dam, the fetuses are heterozygous. They're phenotypically wild type and they express normal levels of CD163. So they're CD163 normal. And then we have, just for the purpose of completeness, we have knockouts. If you take these dams and you infect them at 90 days gestation, this is what you get. In the case of the wild type dam, these numbers refer to different degrees of pathology in the individual fetuses that are taken about 100 days after gestation. And then these numbers down here, that's PRRS viremia. If you have the knockout dam and the wild-type fetuses, there's no infection. There's no infection. And then finally, again, for complete list, no infection in the negatives. So what does this mean? What it means is that this is really the end of reproductive PRRS. If we have a mechanism that can protect the fetus during reproduction from PRRS, then you can think of all those biosecurity measures we no longer have to use, things like checking semen for PRRS, etc. And so really this is the end of reproductive PRRS in my mind. At least we can demonstrate that we can get rid of it. However, interesting, the pigs are born with normal levels of CD163. So they are susceptible for PRRS. So just in case you were worried, we still need that miracle vaccine to carry the pig through the rest of production. And this is just all the people who contribute to this. Again, the pork board, the NEFA ward, and then these are just the people who are in my lab. Thank you very much.